Each year, the Nobel Prize shines a spotlight on discoveries that have profoundly shaped humanity. The Nobel Prize in Physiology or Medicine honors breakthroughs that directly impact our health and understanding of life itself. This year, the award recognizes 3 scientists who unraveled a central mystery of immunology: how our bodies defend us without turning against themselves. Their story is one of scientific detective work, revealing a hidden mechanism that promises to transform medicine for generations. Picture your body as a fortified city, its walls strong and its gates carefully guarded. Within this city, the immune system stands as a tireless army, always on alert, made up of countless cells and proteins working together in harmony. These defenders are constantly patrolling, searching for any sign of danger, whether it's a virus, a bacterium, or any other unwelcome intruder. Just as a city's guards must be vigilant against threats, our immune system is always ready to spring into action, protecting us from harm every single day. The true genius of this system lies in its remarkable ability to tell friend from foe. It can distinguish our own healthy cells from dangerous invaders, launching precise and targeted attacks only when necessary. This careful discrimination is essential. Without it, the immune system could mistakenly attack the body's own tissues, leading to serious problems. Instead, it acts with incredible accuracy, neutralizing threats while preserving the delicate balance of our internal environment. At the heart of this defense is the adaptive immune system, a specialized branch featuring T cells and B cells. These remarkable cells have the ability to remember past encounters with harmful microbes. When the same threat returns, they respond faster and more effectively, forming the scientific foundation for how vaccines work. Vaccines train these memory cells, preparing our immune system for future battles and making us more resilient to disease. But with such power comes great responsibility. The immune system's strength must be tightly regulated, because an overzealous response can be just as dangerous as the threats it fights. If the immune system attacks too aggressively, it can cause inflammation, allergies, or even autoimmune diseases, where the body mistakenly targets its own cells. Maintaining control is crucial to ensure that protection does not turn into self-destruction. Achieving this balance is a delicate act. The immune system must be strong enough to destroy harmful invaders, yet gentle enough to avoid harming the body's own tissues. This equilibrium is vital for good health, and even small disruptions can have significant consequences. Scientists and doctors work tirelessly to understand how to support and restore this balance, especially in people with immune disorders. For decades, one of the greatest challenges in medicine has been to unravel the secrets of this intricate system. How does the body maintain such a delicate peace, allowing us to thrive in a world full of microscopic threats? Researchers are constantly seeking new answers, developing innovative therapies and treatments to help the immune system function at its best. The immune system's command is a marvel of precision. It must direct its soldiers to attack invaders with accuracy, while avoiding friendly fire that could harm the body itself. This remarkable coordination is what keeps us healthy, and understanding it better may hold the key to treating countless diseases in the future. Sometimes the immune system's recognition fails, and it attacks the body's own tissues. This is autoimmune disease. From rheumatoid arthritis to type 1 diabetes and multiple sclerosis, these conditions devastate millions. Traditional treatments blunt the entire immune system, leaving patients vulnerable to infections. Medicine needed a way to selectively control misguided immune responses without disabling the whole system. Scientists suspected a built-in mechanism for self-restraint, a regulatory force that keeps the immune army in check. Discovering this mechanism became one of medicine's most urgent quests. Success would mean safer, more precise therapies and hope for millions. The 2025 Nobel Prize in Physiology or Medicine goes to Mary E. Brunkow, Fred Ramsdell, and Dr. Shimon Sakaguchi. Dr. Sakaguchi, from Japan, identified a special population of cells that suppress self-destructive immune reactions. Brunkow and Ramsdell in the U.S. discovered a gene, FOXP3, whose mutation causes catastrophic immune failure. Their separate paths converged. Sakaguchi found the cells, Brunkow and Ramsdell found the gene. Later, Sakaguchi showed that FOXP3 is the defining marker of these suppressor cells. Together, they revealed the body's internal system for immune control, from cell to gene. Their discoveries are now a cornerstone of modern immunology. Most T cells are immune warriors, but Sakaguchi, Brunkow, and Ramsdell discovered a different kind, regulatory T cells, or TREGs. TREGs act as the immune system's peacekeepers, preventing attacks on the body's own tissues. Sakaguchi's experiments in mice proved these cells actively suppress autoimmunity. Brunkow and Ramsdell's discovery of the FOXP3 gene revealed the genetic blueprint for TREGs. Without FOXP3, the body can't produce functional TREGs, and immune chaos ensues. The convergence of these findings gave TREGs a clear identity and function. This breakthrough turned a mysterious phenomenon into a tangible target for medicine. TREGs are now recognized as the key to immune tolerance. The journey into understanding immune regulation began in 1995, when Dr. Shimon Sakaguchi and his team made a groundbreaking observation. Through meticulous experiments, they discovered that removing a specific subset of T cells from mice didn't just weaken their immune system, it actually triggered the mice's own immune cells to attack their bodies, leading to severe autoimmunity. This was a pivotal moment, as it suggested that the immune system is not only capable of attacking invaders, but also requires active regulation to prevent it from turning against itself. Sakaguchi's work provided the first clear evidence that immune tolerance, the body's ability to avoid attacking its own tissues, was not a passive process. Instead, it was actively enforced by a special group of cells, which he called suppressor cells. This discovery challenged decades of assumptions and opened up a new field of research into how the immune system is kept in check. Fast forward to 2001, when scientists Brunkow and Ramsdell made another leap forward. They identified a single gene, FOXP3, as the master regulator of immune tolerance. Their research showed that FOXP3 acted like a genetic switch, controlling the development and function of these crucial regulatory cells. This discovery was monumental, as it provided a molecular handle to study and manipulate immune regulation. They found that a single mutation in the FOXP3 gene was enough to unleash the immune system's destructive power, causing devastating autoimmune diseases. This finding explained why some rare genetic disorders led to uncontrolled immune attacks, and it highlighted the importance of FOXP3 in maintaining balance. In 2003, Sakaguchi's team made the final connection. They demonstrated that FOXP3 was not just associated with regulatory T cells, it was essential for their identity and function. This united years of discoveries, showing that these cells, now called TREGs, were the body's peacekeepers, and FOXP3 was their defining marker. This cascade of breakthroughs gave science a new vocabulary: TREGs, the regulatory T cells; FOXP3, the genetic signature; and a clear function, suppressing harmful immune responses. The timeline of discovery, from 1995 to 2003, marked a revolution in immunology. Their collective work transformed immune suppression from a vague, theoretical concept into a concrete biological reality. What was once mysterious became measurable, observable, and most importantly, actionable for future therapies. Today, this foundation supports a new era of therapeutic innovation, inspiring scientists and doctors worldwide to harness the power of TREGs and FOXP3 in the fight against autoimmune diseases, transplant rejection, and beyond. The discovery of TREGs and FOXP3 opened new frontiers in medicine. For autoimmune diseases, therapies now aim to boost TREG function, restoring immune balance with precision. In cancer, the goal is to block TREGs that shield tumors, unleashing the immune system against cancer cells. In organ transplantation, enhancing TREGs could help the body accept new organs without harsh drugs. Over 200 clinical trials are underway, exploring these possibilities. The Nobel Committee called this a new handle for medicine, a way to recalibrate our most powerful guardian. The future of immune therapy is being rewritten. The laureates' careers reflect the spectrum of scientific progress. Dr. Shimon Sakaguchi at Osaka University is the persistent investigator who defined the field of immune tolerance. Mary E. Brunkow, now at the Institute for Systems Biology, used genetics to unlock the identity of TREGs. Fred Ramsdell, now in biotechnology, is translating these discoveries into new therapies. Their paths, from academic research to clinical application, showcase the collaborative nature of modern science. Their combined efforts have reshaped our understanding of immunity. This Nobel Prize honors not just individual brilliance, but the power of teamwork in science. The 2025 Nobel Prize marks a paradigm shift in immunology, recognizing not just a single discovery, but a body of work that has fundamentally changed our understanding of the immune system. This honor celebrates decades of dedication, innovation, and the relentless pursuit of knowledge by scientists who dared to ask the hardest questions. Today, immune tolerance is no longer viewed as a passive state, but as an active, dynamic process, one that is carefully orchestrated by regulatory T cells, or TREGs, and the FOXP3 gene. This new perspective has redefined how we approach immune-related diseases and has opened the door to innovative strategies for diagnosis and treatment. The discoveries of Brunkow, Ramsdell, and Sakaguchi are now fueling a wave of clinical trials and experimental therapies. From autoimmune diseases like type 1 diabetes and multiple sclerosis to cancer immunotherapy and organ transplantation, their work is directly impacting patient care and offering new hope to millions around the world. The ability to precisely measure, track, and even manipulate TREGs is transforming both medicine and research. Scientists can now design targeted therapies that restore balance to the immune system, reduce harmful inflammation, and improve outcomes for patients who previously had few options. Their groundbreaking work has not only answered long-standing questions, but has also sparked a wave of curiosity and innovation. New generations of scientists are inspired to explore the mysteries of immune regulation, asking questions that will shape the next era of discovery. The discovery of regulatory T cells is not the end of the story, but the beginning of a new era in immunology and medicine. Each breakthrough builds on the last, and the journey of understanding continues, promising even greater advances in the years ahead. The story of Brunkow, Ramsdell, and Sakaguchi is one of curiosity, persistence, and collaboration. Their willingness to challenge established ideas and work together across borders has set a powerful example for the scientific community. Their legacy will shape the future of medicine for decades to come, inspiring scientists, clinicians, and patients alike. The impact of their discoveries will continue to ripple outward, changing lives and expanding the boundaries of what is possible in science and healing.